All right. Now, you might have noticed on Blackboard, that's what's called, this file says clean at the end of it. That is because we have lots of mnemonics of memory tools in medicine. One of them that I have for the cranial nerves is a little on the dirty side. And I've kind of decided not to really show that one anymore. I found some more G-rated ones, but there are some that are way dirtier than mine. In fact, the one that I was taught in med school was completely disgusting. So I'm not using that one at all. But I still do use a dirty one to this day to kind of help me out. But we're going to tone it down, like I said. So that's why you had the clean version. Why is nothing working tonight? There we go. All right, so the first thing we're going to look at, we're into the nervous system now, the last system that we have to be concerned with for this semester. The file that we had to go over that we're looking at here is not really all that large compared to some of the previous sections. Oh, excuse me, good gracious, where'd that come from? Oh, mute button here. Sorry, cough sneaking up on me. All right, so not as large as some of the files we looked at before, but some of the things are a little more intense, if you want to say it that way, because we have several models that are pretty straightforward. But if we were in the classroom, then probably Wednesday or even next week, we would have been dissecting sheep brains and ox eyes. But of course, we're not able to do that. So I have some videos for you, so you'll be able to look at all those pieces. Some of the stuff on the videos is not necessarily what we need to know. So I've tried to edit this file down to exactly what you need to know for the test sheet, if I remember to put it up this time. So stick to that. All right, now I think that's the right one. I have to look back at that too. All right, so this is the model of the neuron, the action cell of the nervous system. These cells are sending signals so that we can respond internally and externally to our ever-changing environments. Now, I'm not going to ask you what this is. You just need to know that this is the neuron. Now, there's lots of things on here that I have erased. So just what's on here are the things that I will be testing. Don't worry about if you look somewhere and see some other things. So what you'll need to see is all these little gray porcupine little spikes here. These are the dendrites. The dendrite receives the incoming message for the neuron. You need to know about the Nissl bodies, these blue things inside the cell. They are technically rough endoplasmic reticulum. If you put rough endoplasmic reticulum on the exam, I'm not going to mark it wrong. But for the nervous system, if you're going on into a higher science or on into patient care, then you'll be expected to know in the future these are Nissl bodies. Either one's okay right now. This little meaty triangle thing here, this is the only place I can ask this on the model. So this is the axon hillock. Now, I said the dendrites receive the incoming message. The axon, this long thing here, takes that message and sends it somewhere else after it's been interpreted. So it's going to receive the message and send it to a muscle, gland, or wherever else, or another neuron, just wherever else this needs to go. So dendrites get the incoming message, axon acts on other things. All right, so it sends the message out. So most likely we'll ask this from here. This is a little too confusing down here. So this is the axon. All right, so axon hillock is where the axon grows out of the neuron. So this meaty triangle here. But this actual part here is the axon. We're not going to have to know these things right now. If you're in lecture, we're going to talk about these a little more extensively. Just for now, know that the space between these two cells, that's called the node of Ranvier. Or if you're from this area around South Carolina, that's the node of Ranvier. It helps me spell it easier, but technically, if you want to pronounce it, node of Ranvier. I think his name was Claude Ranvier that discovered these things. Whatever, it doesn't matter. We'll talk more about this and what these are and what that is if you're in lecture. Then it will make more sense. All right, so that should be it for that model. The next model will be the, spinal cord. Now, so what we're seeing here is the white matter and then gray matter. White matter from now on in the nervous system, whenever you see white matter, is this stuff here, the myelination. An unmyelinated axon, its transmission is much slower. Myelinated, the axon, transmission is much faster. So we're going to see the white matter on the outside and the gray matter on the inside, that Honda symbol there, a butterfly, whatever you want to think it is. When we look at the brain in a little while, we'll see that the gray matter is on the outside and the white matter is on the inside. So it kind of flips when the brain extends down to the spinal cord. But for now, we don't need to know any of these names out here. That's something that will come later if you're going in that direction. You'll see this split in the middle. The spinal cord is technically a mirror image of itself, so two separate pieces that communicate to one another. So you'll need to know anything that is a slight sunken in area is a sulcus. So a sunken sulcus in the front, there's a much bigger crack, and that's a fissure. So this is a couple of ways to understand front from back. So the back has a very narrow indentation, but the front has a big crack. And also, if you look at the butterfly, Honda symbol, whatever you want to call it, the larger wings are in the back, smaller wings are in the front. So the big crack here, anterior median fissure, this is right in the middle and it's in the front, so anterior median fissure. In the back, not quite as deep, so it's a posterior median sulcus. Let's see, yeah, that's all we need to know for now. Now the butterfly, these parts in the back are the posterior horns. We have the anterior horns up front and the little tiny bump on the sides, the lateral horns. All right, in the middle, yeah, we don't need to know this part. There's a part that crosses over the middle. If you're going into patient care, this is something you'll learn a little bit later. This will be the gray commissure, where the transmission signals can cross over to let both sides of the spinal cord in the brain know what's happening. But what you do need to know, there's a black dot right here. That is the central canal. So we're going to see that cerebrospinal fluid in just a little while. So it's traveling throughout the brain and down through the central canal to feed and nourish the spinal cord. All right, so now the parts that are going inside and outside of the spinal cord. So towards the front, these are the ventral roots, also called the anterior roots, so ventral or anterior. Back in the very first section, we were looking at anterior versus posterior, and I told you that later we would talk about ventral and dorsal. So ventral and dorsal mean the same as anterior and posterior, except we usually see ventral and dorsal in quadrupeds, four-legged animals. But for some reason, the anatomical community, whatever you would consider that to be, the academia, can't really decide whether or not we should call this anterior ventral. So I think there's a push to switch these words. But for now, these are anterior roots or ventral roots. Either one's okay. Then in the back, this on the exam will be a feature, and this will be a structure, and this will be a structure. So this feature is the dorsal root ganglion, or ganglion if it helps you spell it. So when all these signals are coming into the body, a lot of times, if you're familiar with speaker wire, I used to run a lot of speaker wire in cars and houses. It was a lot of fun. But you'll know that over time or over space, the more speaker wire you have, once you get out to the end of that wire, the less the signal is going to be. It decreases over time. Same thing happens with the nervous system. If we're sending a signal down from the toe up to the spinal cord, that signal can decrease over time. So once it gets to here, this is a collection of nerve cell bodies. So it will deliver its message here, and this will send out a new message that's stronger. So dorsal root ganglion is just this collection of cell bodies or nerve cell bodies that will help push this signal along. That will run into the structure, dorsal root, or you can call it posterior root. Either one will be okay. All right. So as we're going to see here in a little while, we're going to see that this is the afferent neuron as it arrives at the spinal cord. This will be the efferent neuron as it exits the spinal cord. I'm not going to ask you that from here, just trying to get you to understand what you're looking at here. So if you were to stick your hand over a hot stove, then that signal will come in through here, and here to be interpreted to say, ouch, that's hot, and the signal will come out through here to tell you to pull your hand back. So we're going to see something like that in a second. I just wanted you to kind of get the idea with the actual model here before we get down to that picture, because the other part is just a picture. Okay, moving right along. Yes, we didn't even notice. So there's lots of different types of neuroglia. Technically, neuroglia from Latin means nerve glue. These are all the little cells that support the nervous system, basically hold the nervous system together. Lots of them on here, but I've marked in blue the only ones that you need to know. And these three are going to be fill in the blank questions from the words that you see, or the exact wording that you see here, right? No. No. See, there's another piece of this that's supposed to be there that I thought I had updated, but apparently I did not. All right, so let's just start from way up here at the top. Now the model we looked at first, the neuron, this one here, these white things here are these Schwann cells. You need to know that the Schwann cells, they myelinate the axon, but they're only found in the peripheral nervous system. The central nervous system is the brain and spinal cord. Peripheral nervous system is all of the other nerves around the body. So Schwann cells are only active in peripheral nervous system. The oligodendrocytes are only active in the central nervous system. I think it's worded like this. If not, then it's going to say form myelin in the central nervous system. But I'm pretty sure on the test, this is the wording. So no oligodendrocytes. Get the spelling as close as you can, and I'll give you credit. But these only do the myelination in the central nervous system. Schwann cells only myelinate axons in the peripheral nervous system. Now the ependymal cells, this is the one I was looking at a second ago that was causing some problems. These cover the inside of the ventricles. The ventricles are the open spaces inside the brain. The ependymal cells come together to form the choroid plexus. Now, what's missing on here that you will need to know, the choroid plexus creates the cerebrospinal fluid, or CSF. So add that in somewhere. I'm going to try, if I can remember, to come back here later and add that line to make sure you understand what you're going to be seeing on the exam. So cover the inside of ventricles. A bunch of ependymal cells together form the choroid plexus. The choroid plexus is the group of the cells, ependymal cells, that draw the plasma out of your blood to make cerebrospinal fluid. So know these three, and I'm going to try to fix this one for you. And, oh, I do have a pen in here. So give me just a quick moment to write that down because I'm going to forget if I don't write it. Okay. Then I'll forget to look at my list, and then I'll further forget to write it. Okay, moving right along. Now we're looking at the brain. Two main parts of the brain. You've got the cerebrum, the largest part there, and the smaller part back here is the cerebellum, which means little brain. You'll need to know the lobes of the cerebrum. So up in the front here, this is where the face would be. So this would be the frontal lobe. Up top, the parietal lobes. In the back, occipital lobe, and on the sides, the temporal lobe. So the same name as the parts of the skull. Keep moving there. Now this odd looking picture, if we were in the classroom this semester, we were cutting the brains, this is the first thing that we cut off. You're going to see this exact picture on the test. I'm going to ask you to identify that structure. That is the dura mater. That's all you need to write. But that is that tough, durable meningeal layer that covers the central nervous system. So it surrounds the brain, surrounds the spinal cord. And as I said, it's tough, and that's why it's called dura, because it's very durable. So with our scissors, it would be kind of difficult to cut through this. It's something you'd have to actually touch and play around with to fully grasp or understand. Of course, if you're going on into patient care, you'll have plenty of other opportunities to cut things up. So you will come in contact with this again. Just for now, this semester, when you see this on the exam, this exact picture, I'll ask you to identify the structure. That's the dura mater. Now there's take that picture off, expected a different picture next. There are a couple more layers, but I'm not going to ask you about those, just as the main one that you need to know about for now. In lecture, we'll talk more about it, but superficially is the dura. Beneath that would be arachnoid, and beneath that would be the pia. And then we'll talk more about that in lecture. Now this model we're going to see from two different angles, right? Yep. These are the ventricles of the brain, the open spaces within the brain. And we'll see here, this one that goes all the way around here is the lateral ventricle. You're probably going to see it on the exam like this. So get to know the lateral ventricles like this. Third ventricle looks like a little bird head with the eye and the beak there. And yeah, we'll leave it like that. And then the third ventricle is connected to the fourth ventricle by the cerebral aqueduct. So a couple of ways I can ask this structure here, either from this model or from the brain model, or that brain dissection we're going to look at. All right, so coming back over here. I see this is more difficult to see this way. So if I were to ask cerebral aqueduct from the model, it would be at this angle. Fourth ventricle would be from this angle. This pink paint that you're seeing here, and you can't see it, it's underneath these as well. That is the choroid plexus we just looked at. So made up of the ependymal cells. Inside these ventricles is where the cerebrospinal fluid is made from your plasma, constantly circulating to keep everything clean. All right, so those are the ventricles. Now we're seeing the model of the brain, and I put it right next. No, I didn't. Where did it go? Great. Usually, I'm going to have to go in and add that picture as well, because that picture is supposed to be there. I don't know why this file's messed up like this. Let me make another quick note so I'll remember. And I was going to look over this file before we got started, and I thought, why not? Because I've already have everything exactly like I like it, but apparently I don't. So when you come back to this, you're going to see the brain model with the brain dissection on the next page so you can compare the two. I think I might have taken this out last semester because since we're not going to be dissecting, it's going to be very difficult for me to show you some of these things and be able to fairly test on them. I'm kind of wondering if that might be the best decision for this semester as well. So the video should already be uploaded for you to look at. If not, I'm gonna make sure it is uploaded. But yeah, looking at this right now, the only thing I could think of from a dissection as far as the brain is concerned, it would be fair, is the dura mater, since I don't have that other picture. So let's just move on from here. Yeah, that's fine. All right, so we have the cerebrum once again. Cerebellum is in the back. This big white band here that connects the right and left hemisphere, that is the corpus callosum. All right, underneath the corpus callosum, there's an open space that is usually here. That is where the lateral ventricle is actually found. And you can see a bird head down here. So this would be the third ventricle. I'm not going to ask third ventricle from this model, but I may ask, I probably will ask, the cerebral aqueduct, and can also ask fourth ventricle. So know these two here, cerebral aqueduct leading to the fourth ventricle. All right, also under the corpus callosum here, this round structure, this is the thalamus. The little dot that makes up the eye on the ventricle model, that is the intermediate mass of the thalamus. So watch the questions very closely on the exam for this part. The arrow is probably going to be pointing at the same place, but if the question says identify the structure, it's thalamus. If it says identify the feature, it's intermediate mass. All right, the space just below the thalamus is the hypothalamus. Makes sense. Hypo means below. So hypothalamus extends down here. All right, I don't have that listed, which is pretty good. This little bump back in here is the mammillary body. And then coming back down around this way, we have this big pregnant bump there. That is the pons. And kind of behind the pons is the medulla oblongata. And that is going to become what was erased down here. It's going to become the spinal cord. I'm not going to ask spinal cord from this model. That's why it was erased. But it's noted the pons and the medulla oblongata are part of the midbrain or brain stem that will lead to the spinal cord. Now, as I said, we have the cerebellum here. So if I ask you from the outside to identify the structure, it's cerebellum. The white matter inside the cerebellum is a feature of the cerebellum. So this, if I'm pointing inside here and ask you to identify the feature, that is arbor vitae. From Latin, that means tree of life. Don't write tree of life, write arbor vitae. So structure from the outside is cerebellum. Feature on the inside is arbor vitae. All right. The only thing that I don't know how I missed that, but it's the only thing that I haven't covered. This little bit of tissue back in here is the pineal body or pineal gland. And you'll learn more about that if you're going on to anatomy 2. All right, we'll see hanging down here is pituitary gland. I'm not going to question that on the exam, but you'll need to know all about that when you get into anatomy 2. So kind of know where it's located. That's kind of hard to see in this picture. There's a couple other things that aren't listed on there. Now you do need to know the cranial nerves, the 12 cranial nerves, and this is where that dirty mnemonic used to be here. So the 12 cranial nerves in order, there we go. So the olfactory starts off at the front of the brain there. So olfactory is used for smell. So olfactory nerve is cranial nerve number 1. Then to optic, which is for seeing, oculomotor, which is for moving the eyes. Trochlear also have a bit of a function with the moving the eyes. Trigeminal is a very large nerve root. We've looked at the supraorbital notch and the infraorbital foramen and the mental foramen. So the 3 branches of the trigeminal nerve come out through those holes to innervate the face. Then we're going to have the abducens, which will also have a little bit to do with moving the eyes. The facial nerve deals with the facial muscles of expression. I should say this too, I'm saying what all these do. You're not going to need to know for lab exactly what every one of these do. You're going to need to know very basic sensory motor or both, but just kind of giving you an idea of what's going on with some of these. Kind of giving you the in order as well. So we have the vestibulocochlear, which is involved with hearing and balance. Glossopharyngeal, which deals with the tongue and the pharynx. The vagus nerve is one we're going to talk about a little more extensively in just a moment. It's cranial nerve number 10. We got the accessory, which is kind of thought to not really be part of the cranial nerves, but we're not going to argue that tonight. Hypoglossal, again, with part of the tongue and some of the pharynx. So here is a couple of mnemonics that I found that were a little more user-friendly for the classroom. So choose one of those if you like. If you don't like those, then you're welcome to go online and look up medical mnemonics, cranial nerves, and you'll probably see some of the ones that I'm referring to. So if you have spare time, I'm not saying you have to. Just know that if you look up some of these mnemonics that we use in medicine, they're going to be dirty. All right. Next is going to show us in order whether these cranial nerves are sensory, motor, or both. All right, so these, like I said, going right in order. Some say marry money, but my brother says big brains matter more. And there are dirtier versions of this as well. But if we were to say, what's cranial nerve number 3 on the exam? And you're going to say cranial nerve number 3 is oculomotor. And I'll say, well, what yeah, give the function of or the function of cranial nerve number 3. Then you'll use this mnemonic to know that cranial nerve number 3 has a motor function. All right, so keep that in mind when I'm asking that on this exam for lab. Whenever I ask for the function of the cranial nerves, I'm only asking for sensory, motor, or both. Don't give me movement of the eyes for number 3. Just motor is good enough. And this one is probably something you should work on a little bit every day. Because if you go a couple of days without trying to memorize these, you're going to forget them pretty quickly, unless you have a really good mnemonic. All right, moving on. Let me check real quickly here. What is this thing? There we go. Okay. Now, for some reason, I didn't see that anyone was really connected here. So if you have any questions, I'm going pretty fast. Feel free to jump right in. This thing is not telling me if the volume's working. And now I just got rid of everything. What is my computer doing? Okay. There we go. We're back now. All right. So now we have the vertebral column. I might have to kill kids in just a moment. Okay, they went away. So we have the vertebral column here. So you remember when we're looking at the bones of the vertebral column, we had 7, 12, and 5. We said that was breakfast, lunch, and dinner. So we have that same thing now, except for with spinal nerves, you get to sleep in an hour later. So now it's 8, 12, 5. So no change in the rest of them. Still about 5 here and still about 3 down here. But as far as spinal nerves, I'm only asking for cervical, thoracic, and lumbar. And you're going to have a better model than this. And all you'll need to do is count those yellow things. And what I'm going to do is I'm not going to put it in the fringe area where it could be cervical or it could be thoracic. I'll be sure it's in the middle of one of these regions so that all you have to do is count and know exactly which region you're in. I hope that makes sense. But I don't have I thought I had another picture on here, but I don't. So this is the only model that I have to look at. So when you're looking at this model in the exam, these yellow things are going to be very clear and very easy for you to count. All right, so 8, 12, 5. All right, the brachial plexus, you might have this exact picture, I'm pretty sure. Yeah, it's probably going to be this picture. And all I did was erase the words. So all you'll need to do is identify this. This is the brachial plexus. These are the roots of all these nerves that are coming out that are going to eventually innervate the arm. So that's all this is really, just to basically have this picture and ask you to identify these this area here. So brachial plexus. This is supposed to be the model of a leg. It's kind of difficult to tell in this very poorly done picture, but we have the sciatic nerve that's running all the way down the back of the leg. So essentially from just inside that butt cheek, basically coming out the bottom of the spinal cord, heading all the way down to the foot, that's sciatic nerve. All right. Over here, we'll have the model of the arm, a little easier to tell now. But this one is running right down the middle of the arm, going to the middle of the hand. That is the median nerve. We don't need to know the others right now. This is yellow, usually denotes a nerve. All right, so you'll need to know about the nerve that innervates the diaphragm. Now, I don't have another picture. But what I usually use for this on the exams, we have this model that is clearly of the lungs and the heart and has the diaphragm at the bottom. And I'll usually point to that diaphragm and ask you to identify the nerve that innervates this muscle. And on this test, you're going to see the word innervates quite frequently. So innervate just means to bring nervous stimulation to. So the nerve that innervates the diaphragm is the phrenic nerve, made up of C3, 4, and 5. So if you write C3, 4, and 5, that's correct as well. But phrenic nerve is usually easy enough to remember. All right, now this is what I was talking about earlier with the vagus nerve. This is one of the cranial nerves, cranial nerve number 10. Now you're not going to see this picture, but I have 3 ways historically that I've asked this question. This is the longest cranial nerve. So I'll say on the exam, identify the longest cranial nerve, or I can ask you to identify the only cranial nerve that leaves the head and neck region. Or I can get any of the models from the classroom and ask you, as such, let's say that I'm using the heart for this example. So you'll have the model of a heart and it'll say, identify the cranial nerve that innervates this organ. So that's vagus nerve. And you'll see it innervates the stomach, intestines, liver, the lungs, the kidneys, the reproductive organs to a certain extent down here as well. So the vagus nerve goes all over the body. All right. So now we're going to move on to the eyes. Now this little chemical equation down here may be helpful to you. You will have probably a multiple choice question that's going to ask something about which cranial nerve controls which muscle of the eye. This chemical equation I've used forever, and it's saved me many times on exams. But it's LR6SO4AO3. We're going to look at what that's talking about now. So the muscle directly on top of the eye is the superior rectus. The one directly on the bottom of the eye is the inferior rectus. Now, whether you have medial or lateral is sometimes a little bit of confusion. So what you're looking for here is this tendon. The tendon always points to the nose. And on the nose, we have the medial rectus. On the lateral side of the eye, of course, is the lateral rectus. So use this as your giveaway. Whenever you see this little tendon sticking out, you know that you're on the nose side, which is the medial side. All right, so wrapping around the top of the eye, the superior oblique, and hanging off the back side of the eye is the inferior oblique. So what this means down here, the lateral rectus is controlled by cranial nerve 6. So that's this one. Superior oblique is controlled by cranial nerve 4, and all of the others are controlled by cranial nerve 3. So for this multiple choice question, it may be patient case based. So I could say a man was struck on the side of the head, and now he has difficulty laterally adjusting his eye, and you determine that the lateral rectus has been damaged. What cranial nerve controls this muscle? So you'll need to know that's cranial nerve 6. All right, let's see, what else do we need to know? Yeah, we need to know these other parts too. Yeah. So as far as the layers of the eye, there should be 3 layers that we need to know, and then do see them. So this outer part, the white covering, that is the sclera. And I'll say on this, identify the layer. Same thing here. If I ask you to identify this layer of this darker color, that's the choroid, this word up here. The CH is a little bit, throws you off a bit. Almost it feels like it's say choroid, but it's choroid. And usually that's black, not really dark brown. And the innermost part has this kind of tan color. That layer is the retina. So it has sclera, choroid, and retina on the innermost part. Now we'll look at the different parts of the eye. So this little glass-looking covering of the eye, that is the cornea. Now the colorful part of the eye is the iris, and the hole in the middle is the pupil. Pupil is usually a feature that I ask about on the test. And a lot of time, no, they're gone, so we're not going to talk about that either. Okay, I keep expecting pieces to be there and they're gone. All right, now there's 2 distinct compartments to the eye. If we cut into the eye, we're going to see this hard marble. That's the lens. Everything from the cornea to the lens is the anterior compartment, or anterior chamber, or anterior cavity. Inside this cavity or compartment here is the aqueous humor. It's very watery. If we were in class, I would tell you to be careful with that, because when you cut open the eye, it usually sprays out. It can get all over you, makes you smell nice. Now, everything behind the lens is the vitreous humor, but everything, the vitreous humor is found inside that compartment and will probably look like this little glass thing here. But everything from the lens back behind there, that area, I think I'm calling it a region, that is the posterior compartment or posterior cavity or posterior chamber. Any of those terms are okay. But just note the structure that fills that cavity is the vitreous humor. So the vitreous humor is like it kind of sounds nasty, but a big ball of jelly, almost like a big ball of snot, really. The watery stuff to the front here is the aqueous humor. I don't really have a way to test aqueous humor for lab. For lecture, it's easier, but for lab, this stuff evaporates very quickly. But for the model here, we have the glass ball that I can ask you about. So that's vitreous humor or vitreous body. All right, we don't need to know the ciliary artery. The optic disc is probably good for you to know. So the optic disc is the structure of the back. So if I'm asking for layer, it's retina. If I'm asking for structure, it's optic disc. Now what we're looking at down here is if we were to slice off the front of this eye and look at the back side. So these red looking things back here, that's the ciliary muscle or ciliary body. Either term is okay. But their job here is to help contract the iris, and that changes the size of the pupil. And we'll look at how that works or what that's called in just a moment. Now we're going to look at the ear. The ossicles are the smallest bones of the body. They are inside the ear. I'm not going to ask you to identify ossicles, but you'll need to identify each of these bones. And this is probably the best picture you're going to see because the ones from the model are not that great. So this one, it kind of somewhat looks like a hammer with a big end down it, or a mallet. That's the malleus. This one, it looks like an anvil. This is the incus. And the one looks like a stirrup from a saddle. That is the stapes. So malleus, incus, stapes. All right, moving on here. So the outermost part of the ear, the part that we see on each other, that is the auricle. If you're going on to anatomy 2, know that auricle is probably the best term for this. This is also called the pinna, so that would be correct. But I suggest that you learn auricle because you're going to see, if you're going to anatomy 2, on the heart, there's some structures that looks like an elephant ear, and those are auricles. So keep that in mind. That might help you remember for next semester. All right, the ear hole, sorry, ear hole, external auditory meatus or external acoustic meatus, either one is okay. Now we get all the way back here. We're going to run into what you would call the eardrum, but you're not going to write eardrum on the test. You're going to write tympanic membrane. All right, we'll look at all the rest of this in just a second, but you need to know this part too, and this is the only picture I have of it, I believe. Well, there we go. This is called the auditory tube. Not a great term for it because it doesn't have anything to do with hearing. The other term, well, this is the easiest term because the other terms are the eustachian tube or the nasopharyngeal tube. So auditory tube's fine. But just so you understand, I'm not going to ask you what it's for, but this kind of helps to equalize the pressure in the middle ear. So an increase of pressure, we can release some air or fluid through here down into the back of the nose. All right, nothing more to see there. So we'll look at this. Now this structure you're going to see, this is the entire balance and hearing apparatus together. This part has been removed. Usually looks like a snail shell. All right, so that snail shell looking thing, that is the cochlea. The cochlea is connected to this long part here, which is the vestibule. That would be that. No, it's the only pictures I have. Yep. All right, so come on. The cochlea is there. The vestibule will be here. And then these 3 rings, you'll see there's an X, Y, and Z axis. These are the semicircular canals. And you're not going to be asked individually, be asked as a group. So semicircular canals. Now inside the cochlea, I don't see this on here, so you might not, you don't have to know it. But organ of corti is going to be found inside of here. That's what helps us be able to detect sounds. But the next thing you'll need to know is this yellow trunk back here. That is the vestibulocochlear nerve. You could be asked like this. That is cranial nerve number 8. But you may also be asked in the long form. I could just ask you to identify the cranial nerve that is involved with hearing and balance. So vestibulocochlear nerve. Okay, what else do we have? All right, looks like that is it for the hearing. Now, I didn't see, did I? Nope. The rest of these ossicles were removed. So I don't believe, unless I do them this way, I don't think they're going to be on the test. Usually I have them listed better than this if they're going to be on the exam. So even though we talked about these, don't expect to see them on the exam. All right, this picture you will see on the exam, the exact picture, actually, a whole thing. And this will be circled, and those words won't be there. But I'll say identify the structure. You'll just write olfactory sensory neurons. So you probably recognize all this. This is the cribriform plate, the olfactory foramina. This is cranial nerve number 1, the olfactory nerve. So those nerve fibers pass through the olfactory foramina. That's how we detect smell, which you'll see there. So identify the structure, olfactory sensory neurons. All right, so this is a reflex test table. Now you need to know these 2 columns, backward and forward. Okay, I'm not going to ask anything about the normal response. This is just here to help you understand a little bit later. But on the exam, I could ask you what nerves are tested with the biceps reflex. So you'll need to know C5, C6. Or I could say, what is the name of the test for C5, C6? You'll need to know biceps reflex. And yeah, I have a way to remember that too. This one, L2, 3, 4, keep the knee off the floor. That kind of helps you remember that a little bit. But the Achilles or ankle reflex, S1, S2. So if you look over to here, now there's a couple we don't need to know, right? Yep, we don't need to know that last one. But for these first two, it's just like the 1-2 buckle my shoe thing. So look over that a bit and it may help you memorize the previous chart. All right. I was talking about this a little bit earlier with the ciliary body moving the iris to adjust the size of the pupil. Now, this is the pupillary reflex. On the exam, I'm not going to ask you how it works or anything like that. The only thing I want to know is what happens when you shine a light in someone's eyes. Normally, they should dilate. Sorry, no, I'm saying it backwards. I was about to tell you something different. Normally they should constrict. If you shine a light in someone's eyes, the pupil should get smaller. Now, what I was trying to say is that every semester I have a big group of people that mess this part up, and it's really easy. What we're trying to do here is adjust the size of the pupil to allow just a certain amount of light in, just enough light so we can see what's going on, but not so much that the retina is damaged. So when you shine a light in someone's eye, that pupil is going to get smaller to prevent the retina from being damaged. So I'm either going to ask you what happens when you shine a light in someone's eye, which you'll say gets smaller, the pupil gets smaller, or I'll ask you the name for the test of shining the light in someone's eye. That's the pupillary reflex. So get used to seeing this. You don't really have to write pupils constrict unless you want to, but just say that pupils get smaller. That's probably the easiest way. And if you're forgetting this way this works, if you have access to a bathroom or a small room with a mirror that you can control the amount of light, just turn the lights off, maybe close your eyes for a moment or two, and then look into the mirror and turn the lights on. And very quickly, you should see your pupils at the same speed getting down to the same size. It happens very quickly, so you have to be ready to watch for it. All right, so this is the reflex arc. Now, I was going over earlier when we were looking at the spinal cord model about putting your hand over the stove. So same thing is happening here. I just wanted you to understand it by looking at the model. But this is the picture you will see with all the words erased. And I'm just going to put an arrow pointing at one of these three and ask you to identify the structure. So if you remember the posterior root or dorsal root, that one's coming into the spinal cord. So it's arriving. That's the afferent neuron. So on the exam, if I ask you for this structure, afferent neuron or sensory neuron, both are okay. The one that exits the spinal cord is efferent neuron or the motor neuron. Right in between the two, the interneuron. So those are the only three things you need to know from this picture. Oh, wow. I expected a couple more slides, but we don't need any more. All right, so that was easy. But that's all that we need for this last test. Kind of taking a little bit easier on you since the first part was kind of brutal with a lot of information. So this isn't as bad, but I would say that you spend most of your time getting to know those cranial nerves. I'm going to post the videos for you, though I don't believe there's going to be a whole lot of questions about the dissection since we can't get our hands on those dissections. But if you need anything, let me know. This was the last class that we need to have. If you need to look over anything again, just let me know and I'll work something out with you. So it was fast. It was a lot of fun. So go ahead and start getting the last test ready for you. And we'll probably put it up as soon as I can so you can have all this time to just take the test whenever you're ready. All right. Have a good night.